Hello class, this is chapter 3 lecture for Bio 101 Anatomy and Physiology, Introduction to Anatomy and Physiology. In this video we're going to be talking about cells and their structure and function. So if you think about the order of things we've talked about so far in this class, we're moving up those levels of organization. So we started with, in the last chapter we started with chemistry, and chemistry is the basis of biology, as we said. It's all of the atoms and the molecules that come together to form living things. Now we're up to the next level, which is the cellular level. And remember, the cellular level, this is the basic unit of life. So we're first going to look at the parts of the cell, and then after that we'll look at the cell itself. And then we're going to look at how things move in and out of the cell, and then how cells are able to reproduce. So we'll kind of move in that direction. So the first part we want to talk about is, so what makes a cell? What are the main parts of a cell? There's many different types of cells. We're obviously going to focus on human cells. Human cells are animal cells. There are also plant cells, bacteria cells, and things like that. But there are three main parts of the human cell and animal cells in general that we're going to focus on. These are the three main parts that are found in every human cell, with some exceptions. There's always exceptions. But these are the three main parts. The first part of the cell is the plasma membrane. This is the outer wall of the cell. It's the barrier of the cell. So remember that cells have to maintain homeostasis. They have to be able to carry out all the functions of life. In order to do that, they have to maintain certain conditions. So all of these things we're going to be looking at here are parts of the cell that allow these cells to control the conditions inside the cell that allow them to metabolize and do the other functions that they need to do to survive. So the plasma membrane is the barrier from the outside environment, because the outside environment is going to be different than the environment inside the cell. And because of this, it's semi-permeable, meaning that the cell can decide what's coming in and out of that plasma membrane. And we'll look at the structure of the plasma membrane here in a second. The second big part is the nucleus. Now, not every cell in our body has a nucleus. There are some exceptions there. And some cells, such as muscle cells, are multinucleate. But the nucleus is an important part of the cell. The nucleus is where we find the DNA, and so it's what is controlling the function of that cell. In here we call it the control center. That's a good way to think about the nucleus. And then finally we have the cytoplasm. The cytoplasm is everything else in the cell. It's kind of this liquidy, gelatinous type of material inside the plasma membrane. So the nucleus is floating around in the cytoplasm. So you have the plasma membrane around, the cell is full of cytoplasm, and within that cytoplasm you have these different things called organelles, the nucleus being one of them. So organelles are what they sound like. So inside our body, as a large organism, as a fully functional organism, we have organs that have different functions inside our body. Inside the cell, we have these things called organelles, which are little organs. And they do a similar type of thing, except on the cellular level. They have different functions that help to carry out the functions of the cell as a whole. And they're just floating around in the cytoplasm. And then we have the cytoskeleton, and that's what it sounds like as well. So it's a skeleton of the cell. It's all these proteins that are inside the cell that give it its shape. They hold organelles in place. They help the cell move and carry out all these functions. So here is an actual picture of what a typical animal cell looks like. This is taken under a microscope. It's been dyed so that you can see the different parts. So all this outside edge is going to be the plasma membrane. The plasma membrane. And then the big thing in the middle, this is the nucleus. Within the nucleus, there's the nucleolus, which is kind of this dark spot within there that has a specific function within the nucleus. And then everything else here is going to be the cytoplasm, with organelles scattered throughout the cytoplasm. Within the nucleus, we have the DNA. In this case, it's called chromatin. And that just is when the DNA is kind of scattered within the nucleus. It's not condensed. We call that chromatin. It's like a ball of yarn that's all just kind of wrapped around inside the nucleus. We call that chromatin. Here's a drawing of what this cell, the different cell parts, so we can differentiate a little bit more all the different parts. I know there's lots of words on here, but I want to point out kind of the main ones that I want you to learn. So the plasma membrane, we already talked a little bit about that. That's this blue envelope here. Okay. The nucleus over here, you can see this big purple nucleus. Notice that there are pores in the nucleus, and that's what allows things to move in and out of the nucleus. There's some things that have to move in and out. The DNA doesn't leave the nucleus, but proteins and RNA can move in and out of the nucleus. And then we have these little yellow dots on the nucleus. These are ribosomes, and ribosomes are scattered on the nucleus itself. They are located on this organelle, which we're going to talk about here in a minute, which is the endoplasmic reticulum. Specifically, this is the rough endoplasmic reticulum, and it's called rough because of all those little ribosomes attached to it. They give it kind of a bumpy appearance. And then there are also ribosomes just scattered in the cytoplasm. So ribosomes are found throughout, and as we go through these different things here, you can see what the jobs of these things are. So the ribosomes, their job is to produce proteins. Next to the rough ER, we have the smooth ER. This is the mitochondria here. That's where the energy of the cell is produced. Here we have the Golgi apparatus. We have some more mitochondria here as well. We have these vesicles, and vesicles are specific organelles that are carrying things among the cell, and throughout the cell and outside of the cell, and we'll look at that a little bit more. We have lysosomes. These are like the digestive centers, kind of the stomach of the cell. They break things down. And then the last thing is just a cytoskeleton in general. And there's some different types of cytoskeleton parts that make it up. There are microtubules, which are these larger proteins. There's intermediate filaments that give structure and strength to the cell. And then actin filaments that help the cell in movement of the organelles in the cell itself. We'll see the microtubules a little bit more in the flagella and cilia here in just a minute. Okay, so I'm not going to go through really specifically all the different organelles, but you will need to know the organelles and their functions. So we'll go over them briefly. We're not going to spend a lot of time looking at those though. So, a little bit more about the plasma membrane, and how it works as a separation. Now, it is built of lipids. If you remember back to the last chapter in chemistry when we talked about organic molecules, we talked about lipids. There's a specific type of lipid called a phospholipid. If you remember looking at this, it was this little, it had like the caricature was a ball with two like legs coming out underneath. And this phospholipid, its structure is very important because it determines the function of this plasma membrane. It's what determines the permeability, in this case being semi-permeable, of this plasma membrane. And the reason is because it has both a hydrophilic and a hydrophobic part of its structure. Again, going back to our last lecture, remember hydrophilic means that it is attracted to water, and hydrophobic means that it is not attracted. It pushes away from water. So most lipids are just simply hydrophobic, but the phospholipid is different because of this phosphate head, this round ball on the top. So the phosphate head itself is hydrophilic. The tails, or the legs, are hydrophobic. So the plasma membrane of a cell has two layers of phospholipids, as you can see here. So one layer and then another layer. The phosphate heads are pointed out and in to the inner part of the cell, which is here, and the outer part of the cell, which is here. And if you think about it, cells live in an aqueous environment. And so the phosphate heads point out because they're attracted to the water around the cell, and they're attracted to the water inside the cell. Now the tails are hydrophobic, and so they are attracted to each other, but they repel water. And so they're pointed towards the inner part of the membrane. So this whole area here is the membrane, the bilayer, and within this membrane we have the hydrophobic tails pointed in and the hydrophilic phosphate heads towards the outside. This is what determines its permeability. We'll look at that a little bit more a little bit later. There are some other things in the membrane as well. These big orange things are proteins, and they're important for a number of reasons. One, because of transport. They allow things to come in inside and outside the cell. They can be kind of like docks for signals that need to go inside the cell. So this one doesn't have a hole through it, but things could attach to this protein and send signals into the cell. There are other proteins that have these are carbohydrate chains. See these little hexagons, these chains of hexagons. So those hexagons are glucose. So these are carbohydrate chains, and their job is to identify what the cell is. Later we'll talk about things like blood type. So blood type, you have a specific blood type, and your cells have that blood type because of these kind of signals, these proteins and carbohydrate chains that identify that cell as a certain type. There are also carbohydrate chains just attached to the phospholipids themselves. And then we have these little purple structures here. Those are cholesterol molecules, and you can see how they're scattered throughout the membrane, and they're supposed to be there. They are what give the membrane its structure. How do I say this? The phospholipid membrane, these are lipids, and lipids are fluid. It's like so it's the consistency, they say, of olive oil. If you can imagine, your cell membranes are the consistency of olive oil. So the cholesterol helps make it more rigid and give it that structure. Okay, and then on the inside you can see some cytoskeleton filaments here that are helping hold that plasma membrane in a certain shape. The nucleus. Okay, as we said, the nucleus holds our genetic information, the DNA. There's the chromatin that we mentioned, which is this conglomerate of DNA, proteins, and RNA all just kind of pushed inside that nucleus. And during certain times, the chromatin will condense into what we call chromosomes. You may have heard of chromosomes before. So humans, for example, have 46 chromosomes. These are pieces of DNA inside the nucleus, separate pieces of DNA, and they condense down into these chromosomes when the cell is reproducing, and we'll see that later in this lecture. We also have the nucleolus. In this case, the plural is nucleoli, and this is the darker part of the nucleus, and its specific function is forming ribosomes. So we saw ribosomes on the outside of the nucleus, in the cytoplasm, and on the endoplasmic reticulum. These are all, sorry about that, these are the ribosomes. These are where the ribosomes are formed. Okay, here we can see the nucleus in here. We already saw a picture of this, so we're going to move through here. The nuclear envelope we saw, so that's the outside of the nucleus, and it has openings in it called nuclear pores that allow things to move inside and out. And the nucleus is continuous with the endoplasmic reticulum. So let's look at the endoplasmic reticulum and ribosomes and how they're all connected to each other. So the function of ribosomes, remember, is to produce proteins. And the way they do that is they use the information from DNA. So DNA holds all of our genetic information. It's what makes us who we are. And remember, in the last lecture, we talked about how we're mostly made of proteins. When you look at somebody, what you see is proteins. The information from those proteins comes from the DNA. So the way that this works is DNA holds the information to make proteins, and the way this happens is DNA is converted into RNA, or it's used to create RNA. So the RNA is a copy of the DNA. The RNA then visits the ribosomes, where the proteins are produced. So the information in the DNA goes through the RNA to the ribosomes, then the ribosomes produce these proteins. So these proteins can be produced different places. One of the places they are produced is the endoplasmic reticulum, and it's part of what we call the endomembrane system. So these are organelles within the cell that are surrounded by membranes, and they're all kind of working together. So the endomembrane system has the nuclear envelope around the nucleus, the endoplasmic reticulum, the Golgi apparatus, lysosomes, and also vesicles. And these guys are all working together. So first, the endoplasmic reticulum. Let's focus on the rough ER. So the rough endoplasmic reticulum is the one that has the ribosomes attached to it. And the reason the ribosomes are attached to it is because it produces proteins. The ribosomes produce proteins that are then produced into the rough ER. Now when that happens, what that means is these proteins are being produced to travel somewhere. So you see all these ribosomes on the rough ER here. These ribosomes are producing proteins that are moving inside the rough ER, producing them into the rough ER. From there, these proteins are going to travel on a path. So here's a picture of this path. Here's the nucleus again with the DNA, the information to build the proteins. Here's the rough ER with the ribosomes attached and the proteins being produced. So if proteins are produced in the rough ER, they will then break off in a vesicle, and this vesicle will travel to the Golgi apparatus. Now, the reason this is called the endomembrane system is because all of these things are surrounded by membrane. It's plasma it's built of the same type of stuff as a plasma membrane. We don't call it a plasma membrane, we just call it a membrane, but it's a phospholipid bilayer. And vesicles, or pieces of this membrane, can move from one place to another because it's all the same stuff. So a piece of membrane can break off of the rough ER, and it can travel to the Golgi apparatus where it can basically fuse with that Golgi apparatus, and then it can move through the Golgi apparatus, and then a new vesicle can form that will be sent somewhere else to do different things. So the Golgi apparatus takes these proteins that were produced in the rough ER and modifies them for whatever function it needs to do. So proteins produced in the rough ER will move to the Golgi apparatus. From the Golgi apparatus, they can then move to the plasma membrane, or they may stay inside the cell and become a lysosome, such as this one here. So here's a lysosome, and then it'll function as a digestive structure within the cell. So that's the function of this endomembrane system as things are moving throughout. We also have the smooth ER. The smooth endoplasmic reticulum is a little bit different. So it doesn't have ribosomes because it's not dealing with proteins. So what it's doing is it's helping to build lipids and types of lipids that the cell needs. It also will work to detoxify substances that end up inside the cell. And so that's the function of the smooth ER. So you can see again, this picture, you have the nucleus, you have the rough ER here. Here you have the smooth ER carrying out its functions, and it's also part of this endomembrane system as well. Okay, we already talked about the Golgi apparatus. You can look through this information here on the slide, but its job is basically to modify those proteins that were produced in the rough ER and then send them to wherever it needs to go. So it's kind of like the packaging receiving center of the cell. Receives these proteins, packages them up a little bit different, and then sends them off. Lysosomes, their job is digestion, as we said. So they're full of these enzymes. Now these enzymes break down organic materials, and so they have to stay inside a vesicle, inside a membrane. Otherwise, they'd start digesting the cell itself, and we don't want that. It's kind of like our stomach, how it holds hydrochloric acid. We've got to keep that inside the stomach. If it gets out of the stomach, it causes all kinds of problems. So the lysosome has a few different jobs. One is to actually get rid of parts of the cell, excuse me, that are getting old. So if there's organelles that are no longer functioning or they're getting old, it will digest those, and then the cell can reuse those parts. It can also digest things that are coming in from outside. If the cell is eating, it's engulfing other things that it's feeding on. And then an example of if the lysosome is not working. So the lysosome has a very important job. Tay-Sachs disease is an example when the lysosome is not working, and what happens is these fats and things build up inside the cell, so the cell can no longer function as it's supposed to, and it leads to the Tay-Sachs disease, the symptoms that we see in Tay-Sachs disease. You can look up a lot of stuff about Tay-Sachs. Here's just a quick thing from the internet. You can see, so these fats and things build up inside the brain and other tissues, and they start to kill the brain cells. It can also lead to other things: cataracts, jaundice, enlarged liver, kidney damage. So very, very damaging disease that's caused by just the lack or the lysosomes not functioning as they should. Okay, this is a little animation. I encourage you to look at this on your slides, and it talks about how lysosomes work and their job. Okay, mitochondria. Mitochondria, these are the powerhouses of the cell. This is where the energy, the ATP, comes from. We talked about ATP in the last chapter. So ATP is the energy currency of the cell. That's what is used to carry out all the functions of the cell. And the mitochondria is where this ATP is produced through cellular respiration. And we talked about this a little bit, but the energy to form ATP comes from what we call cellular respiration. And the main energy source in the cell is carbohydrates, specifically glucose. So as we eat glucose, it goes into cells. Our cells take that glucose and they use oxygen to extract the energy from the glucose, and they attach that to the ATP. And then that energy can then be used to do other things. And the byproducts of this process is carbon dioxide and water. So that's why we breathe out carbon dioxide. And then the water is just used inside the cell. So that all happens inside the mitochondria. The mitochondria is an interesting organelle. It actually has two membranes. Just a side note, the nucleus does as well. So the nucleus and the mitochondria are the two organelles that have two membranes. So not only is it a phospholipid bilayer, but there are two phospholipid bilayers in the mitochondria. And then there's all this folding inside called the cristae, and this helps to increase the surface area of the mitochondria, because this is where the energy is being produced. So the more area that you have to produce energy, the more energy you can produce, and so that's why you see that folding here in the mitochondria. Cytoskeleton. The main parts of the cytoskeleton that I want you to know are the cilia and the flagellum. And these are made of microtubules. So microtubules are a type of cytoskeleton. And these are the two main types of projections from the cell that are formed by the cytoplasm. Sorry, the cytoskeleton. The first one is the cilia, the other is the flagellum. And the way to think about this, cilia are like little hairs, the flagella is like a tail. Generally, cells only have a single flagella. In humans, cells should only have a single flagella, and the only place the flagella is found in human cells is on a sperm cell. No other human cells have flagella. In other animals and things, there's other cells that have flagella. But in humans, the only cell that has a flagella is the sperm cell. And that flagella is made of microtubules. The cilia is found in a number of different cells, and where the flagellum, there's just one flagellum, there are generally going to be a whole bunch of cilia on cells that have cilia. It'll look like it's covered in hairs. And the cilia's job is to move and help things move, either move things along a pathway or to get rid of things. So a couple examples of where we see cilia. So in the fallopian tubes in females, there are cilia that help move the egg along the fallopian tubes. And then also in our respiratory tract, we have cilia on our cells that help to move mucus and other bacteria and things that are getting inside our lungs. It helps move them out of our body. So that's where we see cilia. Here's an example of cilia. This is a microscopic image. You can see all these little hairs. And then these are sperm cells. You can see their flagella. Towards the top here, we have mitochondria that are producing energy that are making this tail move. And that's where we see these in human cells. And here's a nice little chart from your book that gives the different structures in the cell, and it tells what they're made of, and then it also has their functions. This is a good little review of all the different parts of the cell. So please look through that, and you don't need to know a lot of detail. For example, the cilia and flagella, it tells how many microtubules they have. You don't need to know that at this point. We didn't look at centrioles, but know the general functions of each of these. Kind of a fun exercise that you can do is you can compare the cell to like a factory. So if the cell was a factory, what would each of these organelles be inside that factory, for example? So if I was thinking about that, so the plasma membrane, that's kind of the walls of the factory that determine what can come in and out. The nucleus is the control center of the factory. That's where the boss sits and tells everybody what to do. The ribosomes, these are the workers. They're producing a product, which in this case is proteins. Endoplasmic reticulum would be the machinery that the workers are working on to produce these proteins. And then you have the rough and smooth, depending on what product you're trying to produce. The Golgi apparatus is the shipping receiving center of the factory. The vacuole helps to store things. That's a storage room. The vesicle is like a little trolley cart that moves things around the factory. The lysosome, that maybe would be the lunchroom or something like that. The mitochondria is the generator in the factory that's producing all the energy. And then the cytoskeleton, that would be the inner workings of the building that are holding up the walls of the building. So there you go. There's kind of thing to think about as you're comparing these different structures to different things. Alright, that's where we're going to end this little part, and then we will pick up with things crossing the plasma membrane.